recording. So today we're going to talk about Chapter 7, the control of microbial growth. That's largely going to be talking about disinfectants and antiseptics, although antibiotics are also how you can control microbial growth. Anyways, these are topics that students are fairly familiar with and don't have too much trouble with. That's why I talk about Chapter 7 at the very end. And that is, it's a chapter that you guys already have a pretty good understanding of disinfectants and antiseptics. In the lab today, there is no lab. I will be logging in from 6:30 to 6:45 to answer any questions in case somebody has questions. Let me remind you that the unknown notes pages are due on Friday. So if you don't have your last unknown notes up, go ahead and put that up. And then the unknown report is due Saturday. So make sure you turn in your unknown report by 11:59 p.m. on Saturday. The final is going to be Wednesday. You can take it at any time on Wednesday, and it's going to be due at 11:59 p.m. It's an online exam. There is heavy coverage of Chapter 7, 13, 14, and Lab Module 9. These are material that you have not been quizzed on previously, and that's why about 50% of the final will be on Chapter 7, Chapter 13, Chapter 14, and Lab Module 9. The remainder of the quiz will be on the lab and on the material that was covered with quiz 1, 2, and 3, meaning from the start of the book. Any questions about any of that? All right, if no questions, let's move forward. Let me just briefly go to my Clark website and see if there's something I need to talk to you about. I think we're good, but I just want to make sure there's not something I'm forgetting. Stop there and I'll take a look at the grade book. Just because that's one way to look at it all at once. Nobody asked for Chapter 14. I'll make those notes available. And quiz 4A, I need those notes available also. All right. Let me take a look at the grade book, see if there's anything I want to talk about that. The final will be in two parts, a final A and a final B. The final A will be just a few multiple choice or fill in the blank answers. And then, not multiple choice, sorry, what do you call it, essay. And then final B will be most of the final and it'll be multiple choice questions. Oh, there's something I wanted to mention. There is a worksheet to chapter 7. However, it is extra credit. So you can get extra credit from filling in the worksheet to chapter 7. And I remember it's going to be 1 or 2 points. Yeah, all the worksheets to the lab are graded. The infectious disease project is graded. So just the unknown notes, the unknown report, and then the final are not graded. Everything else is graded. Let's see if the total has gotten a little better. Nope, no improvement there. I'll look and see why it's not counting up all of the points that are available. I'm assuming what it's doing is that it's not counting. I think there's like 460 or 465 total points so far. And the total, it's only counting 400 for some students and 415 for other students. And I don't know why the total is different. I'm hoping that the grade book at the end will go up to 720. That's what it should be. And everything will be correct. If not, I will put in the total in the column. I already made it. Total points out of 720, and I'll fill in the percent. But I'm hoping the grade book will get it right at the end and everything will be okay. What I think is going on is that it's not counting some assignments if they are not due. So if it's not due yet, it's not counting the total points. And like I said, everything's been graded except for the final and the unknown notes and the unknown report. All right, any questions? Shut the grade book. Let me start sharing the screen again. All right, let's talk about chapter 7, the control of microbial growth. As always, I show you a slide showing you the major goals for this chapter, as well as a rough outline. Know the terms: sterilization, disinfection, antisepsis, degerming, sanitization, autoclaving, pasteurization, biocide, and germicide. And as you notice, most of these terms are ones you've already discussed or should know. 2, be able to discuss the factors that influence microbial death rates. 3, be able to discuss the 3 main ways in which antimicrobial agents kill or inhibit cells. 4, understand the various types of disinfectants and how they work. And 5, understand the relative resistance of the major microbial groups to antimicrobial agents. When we look at the rate of microbial death upon exposure to an antimicrobial agent, you'll notice that the bacterial populations die at a constant logarithmic rate. This doesn't matter if they're heated or treated with an antimicrobial chemicals. The bacteria tend to die at a constant logarithmic rate. That means that if we plot the death rate, let me blow this up a little. Plot the death rate on a graph, and it's the logarithmic number of microbial survivors on the y-axis, and then the time of exposure on the x-axis. The death rate happens logarithmically on a straight line, meaning 1 log decrease equals a 90% population reduction of cells. Now that's with the log number of cells. If you did the arithmetic rate, it wouldn't be a linear relationship. In fact, it would be this relationship shown here. And what it would be on a graph is when we look at the time in units and the number of survivors, starting with 1,000,000 cells, 1/10 of the cells would survive after 1 unit of time, and then 1/100th or 1/10 of that would survive after 2 units of time, and then 1/3, 1,000th, sorry, not 1/3, 1,000th would survive after 3 units of time, so that there's a constant death rate. Any questions about any of that? Let's shrink that. The rate of microbial death will be affected on the number of microbes present at the beginning of the treatment. The more microbes you have, the longer exposure you need to kill off all of the cells. The fewer cells you have, the less time you need. There are other environmental influences which affect the death rate, such as the presence of organic matter. Usually, hospital specimens will have bacteria present with organic matter. And that could be, feces or blood and bacteria, anything like that. The trouble is the organic matter can inactivate the antimicrobial unit, the antimicrobial drug. For example, bleach is highly inactivated by organic matter. The temperature of the treatment will affect the environmental influence of how quickly the microbes will die. Generally speaking, most microbes will do less well at warmer temperatures than cooler temperatures, meaning if you heat the microbes and then expose them to an antimicrobial treatment, more of them will die quicker than if you cool the bacteria. The presence of the biofilm will also affect the death rate. Generally speaking, if there is a biofilm, the cells will be much more resistant to dying than without a biofilm. As I mentioned, the time of exposure will affect the microbial death rate. Generally speaking, you need longer times for more microbes. You also need longer times if there are spores around. And I'm talking about fungal spores or endospores. Many antimicrobial treatments will not be effective against endospores. That's just something you can plug into the back of your memory. The microbial characteristics, which we'll discuss at the end of this lecture, also affect the microbial death rate. And I've talked about one already, the presence of endospores, that's a microbial characteristic, will affect the microbial death rate. Any questions about any of that? All right, so this table is giving you the different terms, what the definition of the term is, and then comments about how that term's used. Sterilization, the destruction and removal of all forms of microbial life, including endospores, but we don't really count prions because they're almost impossible to kill. Almost, not quite, but almost. Sterilization is something you do to remove common bacteria. And how we do that in the lab is usually we put things in an autoclave. Commercial sterilization is sufficient heat treatment to kill endospores, such as Clostridium botulinum in canned food. We don't use that very much, but the the canned food industry uses that. Disinfection is something we use every day. It's the destruction of vegetative pathogens. When we clean the toilet, we use a disinfectant. We destroy the vegetative pathogens. It may not kill endospores. Antisepsis is destruction of vegetative pathogens on living tissue. Generally, antiseptic chemicals are less harsh than disinfectants, but it doesn't really matter because you can use the same chemical, and in one place you're using it on an object, it's a disinfectant, and that would be like when you use bleach on the toilet. But if you were to use bleach on your cat, it would be an antiseptic. Any questions about that? Degerming, the removal of microbes from a limited area, such as the skin, before you insert the needle in an injection site. You're certainly not sterilizing the skin, that would kill your own cells, and you're not really using antisepsis when you're alcohol swabbing the site you're going to inject. All you're doing is reducing the microbes to a safe enough area, safe enough level that you can now inject. Sanitization is the treatment to lower microbial counts on eating and drinking utensils. And those are used in public health settings, like when you go to a restaurant. They sanitize the plates. They don't sterilize them, but they sanitize them. Any questions about that? A biocide or a germicide is a substance that kills microbes. Biocides and germicides do not usually kill endospores. Bactericide is a special biocide that specifically kills bacteria. Fungicide is another special biocide that specifically kills fungi. And then there's viricides. These are agents which enact viruses. Bacteriostasis, we've already talked about this in the lab, inhibits the growth of microbes. You can remove the bacteriostatic agent and then the growth might resume. So a biocide will kill the microbes. A bacteriostatic agent just inhibits their growth. Any question about that? Sepsis refers to microbial contamination. So asepsis is the absence of contamination by unwanted organisms. And in this class, hopefully you've learned aseptic technique. It is important in obviously microbiology, but also in surgery to minimize contamination of the instruments, the operating personnel, the patient, and the air. Without using aseptic surgery, the patient would likely get infected from the surgery. And if you don't know, before aseptic technique was adopted in the medical profession, most people undergoing surgery got an infection and then actually died of the infection. It wasn't from the surgery itself. All right, antimicrobial agents control microbes by three main ways. They kill or inhibit cells by altering the membrane permeability. If you damage the plasma membrane, the lipids and the proteins in the cytoplasm will leak out, and that will kill the cell. You can cause cellular contents to escape and interfere with normal metabolism, and regardless, you damage the cell membrane, the cell is dead. You can also damage the intracellular proteins, like when you denature proteins or enzymes. Actually, alcohol, how it works is by denaturing proteins. So it's damaging the cell by damaging the proteins. You can also target the disulfide bridges or the hydrogen bonds. Any way you damage the proteins, you will damage the cell and kill the cell. You can also damage the nucleic acids. This will result in mutations, And if you get enough of them, that will kill the cell. Any question about any of that? So the three ways to kill a cell or alter the membranes: damage intracellular proteins or damage nucleic acids. There are various physical methods for controlling microbial growth, and we'll talk about most of these. There's heat, filtration, low temperature, high pressure, desiccation, osmotic pressure, and radiation. Let's talk about heat first. All methods of heat kill microorganisms by denaturing proteins and enzymes, though extreme measures of heat, such as flaming and incineration, turn cells into char. That will also kill them. You can have two types of heat. There's wet heat, such as boiling, or autoclaving and steaming. And then there's dry heat. That would be baking, as well as flaming and incineration. Moist heat denatures proteins and enzymes, causing coagulation of the protein. When you boil a liquid, that'll be at 100 degrees Celsius, or when you're using flowing steam, you kill vegetative bacterial pathogens, most viruses, fungi, fungal spores, within about 10 minutes of exposure. Oftentimes, the cells will die much faster, like if you just expose E. coli to 100 degrees Celsius boiling water, you just need to bring it to a boil, and the E. coli will be killed. Endospores and some viruses may take longer to destroy with heat, and some can survive over 20 hours in boiling water. So like I said, endospores are something that's difficult to kill, and there are not too many antimicrobial agents that kill endospores within a reasonable amount of time. Any question about any of that? All right. So how do we achieve reliable sterilization with moist heat? Well, you use temperatures above boiling temperature, and you use it under pressure. How you do that is with an autoclave. An autoclave is sort of like, if you don't know what one is, it's sort of like a pressure cooker. There's steam under pressure, and it's very effective at killing cells. Increased pressure raises the boiling point that water will boil. It's higher, and the increased temperature and the increased pressure both contribute to killing cells. Steam at 15 pounds per square inch of pressure at 121 degrees kills everything in a liter, or is it a gallon? I think it's a liter. Everything in a liter in 15 minutes. Normally, we run our autoclaves at 15 pounds per square inch and 121 degrees. So you need only 15 minutes to kill everything in a liter. Now, let me state that at atmospheric pressure at sea level, the pounds per square inch is 1. So this is 15 times greater. Any question about that? When you're sterilizing something with steam, not in an autoclave, when you're applying the steam directly, the steam will only kill the surface, and you simply expose the surface of what you want to sterilize directly to the steam. In an autoclave, what you do is you put inside the autoclave what you want sterilized, and then start the autoclave cycle, and it'll build up the pressure and build up the temperature. And in 15 minutes, it'll be done, but you have to lower the pressure gently. So it takes time, and it's usually longer than 15 minutes for the autoclave cycle. Any question about any of that? What is important is that the autoclave will kill endospores in about 15 minutes. All right. Pasteurization was a process started by Louis Pasteur in 1857. It reduces the number of spoilage organisms and kills all pathogens. It's mild heating sufficient to kill organisms that cause spoilage. There are two mechanisms for applying pasteurization. Both of them use denaturation of proteins and enzymes, but you should realize that pasteurization does not kill all organisms. Any thermoduric organism can survive the process of pasteurization. So something like pasteurized milk will not be bacteria-free. All of the thermoduric organisms will survive. It's just that the pathogens are killed off by pasteurization, and a lot of the bacteria are killed, but not all of them. You can pasteurize milk or some other substance, liquid or air, liquid or gas, I should say, for 63 degrees Celsius and apply it for 30 minutes. Or more often, when we have pasteurized milk, it's exposed to 72 degrees Celsius for 15 seconds. So most milk is treated that way, and then it's sold as pasteurized milk. And if you don't know, pasteurized milk lasts longer than unpasteurized milk in the refrigerator, meaning the half-life, how long the milk will last unopened, is longer than a week. And that's why we pasteurize milk. Milk can also be sterilized. This is not pasteurization, it's sterilization of milk, and it kills all organisms in the milk. All you need is 140 degrees Celsius for about 1 second, and this we call ultra high temperature milk. And if you ever see milk in a carton in the store, liquid, or it could be in a can, it does not require refrigeration until it's open because there are no microbes in the milk. It cannot spoil. Whereas pasteurized milk, once you go past its expiration date, it can spoil and will go sour. Any question about any of that? And then, you know, if you don't know, a can of milk, meaning the ultra high pasteurization or in the carton, will last about a year at room temperature. And then they say you should throw it away because not really that it will become contaminated. It won't do that. There won't be bacteria growing in it. But the product itself will not be adequate, the quality of the product itself will degrade. And most of that is by oxidation. There's also dry heat, and we can use this to sterilize, such as sticking something in the oven, killing by oxidation. Hot air sterilization is normally baking. Items placed in an oven at 170 degrees to bake it. You need to bake it for 2 hours to kill all the microbes. Actually, will that kill the endospores? Probably not the endospores. To kill all the vegetative pathogens. So you need to bake it for 2 hours. Did you notice that that is much longer than applying direct wet heat? Generally, you only need 10 minutes with boiling to kill every vegetative thing, meaning maybe not endospores, but almost everything else. And that's because moist heat is much more efficient at transferring the heat to the agent we're heating than air is. Why the cat can climb in the oven. And you know this, you just have to think about it. If you were to put your hand through steam, you would immediately burn it. But we put our hands in the hot oven all the time and then pull out whatever we're cooking. So you can expose your skin to dry heat as long as it's just a short period of time. But you would not want to expose your skin to wet heat for any length of time. All right, any questions about heat? If not, let's move on to filtration. Filtration removes the microbes from liquids and gases as they are filtered through a membrane filter. And the microbes are caught by the pores of the filter and kept on this side. And then the liquid which comes down, in this case, it could be a gas, but in this case it's a liquid, will be filtered and will be free of microbes, the bacteria. This is a very useful way for sterilizing liquids, especially if the liquids would be altered by heating. Some people will sterilize various things we drink this way, like juice or beer. If you don't know, Coors beer is filtered, and that's why it tastes different than a pasteurized beer, which is heated. Some vaccines are filtered. Milk usually is not, because the proteins and the fat of milk would clog the membrane. All the pores in the milk would get, all the pores in the membrane would get clogged by the milk. And so we usually do not filter milk. Low temperature is another way to control microbial growth. Refrigeration from about 0 degrees to 7 degrees Celsius slows the metabolic rate of most microbes, where they cannot reproduce or synthesize toxins, but that may not kill them. Psychotrophs can actually even grow at this temperature, and they can divide about 3 times per day and will reach numbers greater than 2 million within a week, so that the psychotrophs can grow in your refrigerator, and that's why food should be thrown away after about a week. You can cool things cooler, such as putting them in a freezer where the temperature is less than 0 degrees Celsius. That will stop metabolic activity. And we call this slow freezing in a home freezer, as opposed to freezing it with liquid nitrogen, which would be quick freezing. And slow freezing allows ice crystals to form, which can kill some bacterial cells and will kill all parasitic eukaryotic species that we know of. And so if you wanted to make sure that, I don't know, the pork has no parasites in it, you can freeze it, and that will kill the parasites. It would not kill the bacteria cells. In fact, endospores would not be affected by freezing, not be affected at all. But other bacteria like E. coli, you wouldn't necessarily kill all of the E. coli, but you would reduce their numbers by freezing, slow freezing. And then there's quick freezing with dry ice or with nitrogen. That would be putting the substance in the liquid nitrogen. The substance put in the liquid nitrogen freezes so quickly that the ice crystals don't form. And so putting things in liquid nitrogen will not kill most things. Any question about any of that? All right. High pressure can also be used to control microbial growth. High pressure transfers the pressure evenly throughout a liquid, and this actually kills vegetative bacterial cells. It also denatures proteins and alters carbohydrates, killing the vegetative bacterial cells. It does not kill endospores. High pressure alone will not kill endospores, but we can use high pressure to preserve the color, the flavor, and the nutrient value of juices. So oftentimes fruit juices are often pressure treated to preserve them so that they do not go bad. So a lot of the juices that you buy on the store that are in a package and you don't have to worry about refrigerating until you open it, those are usually pressure treated. If we were to heat those juices, which would be another way that you can treat the juice, it would change the flavor. Any question about that? Desiccation and osmotic pressure is another way we can control microbial growth. And these two ways have been used for millennia by people to preserve food. Desiccation is drying. This will prevent microbial growth. The cells can remain viable for years, particularly if they have an endospore, but they will not grow if the item is desiccated. And some examples of desiccated food include drying figs or dried fruit. Osmotic pressure is very similar, where you use high osmotic pressure causing plasmolysis. And this could be from a high salt concentration or a high sugar concentration. And something that's put in something with high osmotic pressure will have its water leave the cell and go into the high osmotic environment, causing plasmolysis of the cell, where the plasma membrane pulls away from the cell membrane. Some examples of food which are preserved by osmotic pressure include molasses and honey. I think I mentioned earlier that my mother had a bottle of molasses that we figured was at least 30 years old. And it never got contaminated, and bacteria, I'm sure, fell into it because of high osmotic pressure. There was so much sugar outside the cell that the water from inside the cell flowed out of the cell into the molasses, preventing the molasses from being spoiled by the bacterial growth. Another example of this would be honey. Honey tends to not go bad because of high osmotic pressure. Any question about any of that? All right. Some the last couple of physical methods for controlling microbial growth include radiation, which damages DNA. You should know that not all radiation damages DNA. Strictly speaking, infrared, visible light are on the electromagnetic spectrum, and the only types of radiation on the electromagnetic spectrum which actually damage cells would be a very low wavelengths like gamma rays and X-rays and UV light. Visible light itself does not damage cells, damage their DNA. And then infrared and microwaves do not directly kill cells, but microwaves tend to heat things up, and that may kill the cell, the heat itself. Any question about any of that? All right. So I mentioned radiation can kill cells. There are two types of radiation. Ionizing radiation, which has so much energy that the ionizing radiation is absorbed by an atom and absorbed by the electrons of an atom. And that electron is going to get so much energy from the ionizing radiation that the electron will be lost from the atom. Let's see what I want to say about that. When the electron is lost from the atom, that means the atom is now an ion. It'll have a positive charge. And ionizing radiation causes ions, which then do damage to DNA. Ionizing molecules can also make molecules of hydroxyl radicals. And we'll talk a little bit about that, meaning ionizing radiation can hit something like water or another molecule, create ions, and then create hydroxyl radicals, which can then go and damage the DNA. Ionizing radiation is used in the food industry, the pharmaceutical industry, and the medical supply industry. So if ever you pick up a package of the sterile Band-Aids, they've probably been sterilized by ionizing radiation. And how I can say that is a Band-Aid is kind of made out of plastic, so you obviously can't heat it. It would be difficult to put it under pressure, although that would work, at least killing the vegetative cells. You could treat it with a antiseptic or disinfective agent, but the trouble with that is Band-Aids tend to have a sticky area and then a cloth area for putting on the wound. We don't want those to become wet. Anyways, ionizing radiation is used with some food, and you usually see that with the backpackers' food. Like they have ionizing food that may be dehydrated as well. And you just add water and then start cooking it. Anyways, a lot of that food is sterilized with ionizing radiation. Another thing is if you go in and you see some meat that has no other preservatives in it, like a meal, and it's just sealed, it's been exposed to ionizing radiation. And then they just put it on the shelf and it will be good for a long period of time, 6 months or a year, because of the ionizing radiation killing all of the microbes within the package. I'm not suggesting you eat this, I'm just explaining it, okay? Another form of radiation which can do damage to DNA is non-ionizing radiation, and this is ultraviolet light, or also called UV light. When UV light hits the DNA, it doesn't cause ions, that's why it's not ionizing radiation. It causes thymine dimers, where the adjacent two thymines, instead of, well, actually, normally the two adjacent thymines don't bind with each other, except in the sugar phosphate ribose, sugar phosphate bond in the single strand. Anyways, two thymines will have a thymine dimer. That's two covalent bonds linking them together. These two thymines will not bond with hydrogen bonds to their corollary A's. And this will create a blip in the DNA, which is one of the ways that some repair enzyme can see this and then remove it and then fill it in again to correct the damage. Let's see, is there anything I want to mention about that? The body can correct it, excuse me, as long as the correction happens before the DNA replicates. Now, another thing is this thymine dimer is very good at stopping DNA replication. The polymerase will not go further. And it also stops the polymerase from transcribing the DNA. So this damage from the ultraviolet light will stop DNA replication as well as transcription. Any question about any of that? All right, these tables show you the different methods you can use, physical methods for controlling microbial growth, and you can look through them as you desire. I thought we talked about that. I did talk about microwave radiation. It's not really good at killing cells, so it's not an antimicrobial agent. However, microwaves tend to heat things up. And if you get enough heat, you can start killing things with the microwave. Anyways, that's physical methods for controlling microbial growth. There are also chemical methods for controlling microbial growth. You should understand that the chemicals we use for controlling microbial growth, there's actually few of them that achieve sterility. Most chemical agents simply decrease the microbial numbers to safe levels or remove vegetative pathogens, vegetative forms from objects. They do not, most microbial chemicals do not remove endospores, although there are a few that do remove endospores. So the principles for effective disinfection, meaning destruction of vegetative pathogens. To have an effective disinfectant, you need to have the right concentration of the disinfectant. Generally, more disinfectant, more concentrated is better, although that isn't always the case. Like when we're working with alcohol and you increase the alcohol concentration, you'll get better killing up to about 70%. But once you go beyond 70%, the killing starts decreasing. And it's very easy to make 95% alcohol because you just boil it and your purity of the alcohol will be 95%. The other 5% is usually water. And 70% alcohol is better at killing cells than 90% or 95%. The reason is that the alcohol is better at getting in the cells, the 70% alcohol, than the 95%. So as I stated, concentration of the disinfectant is one factor to how effective the disinfection will be. The time will be a factor in how effective the disinfection is. Generally speaking, you'll get better killing with longer exposure time of the disinfectant to the microbial agents. pH will affect the effective disinfection. Generally speaking, most disinfectant chemicals work better under an acid condition than a basic or neutral position. Now, obviously, this will depend on the chemical. There are some, a few, that work better under alkaline conditions than acid conditions, but most generally work better under acidic conditions. So that's the one you should remember. And then I mentioned it before, the presence of organic matter can affect the effective disinfection. If organic matter is present, many agents like bleach will start to become inactivated by the organic matter. And then the disinfectant will have less killing. And I'll point out that most clinical situations will have organic matter in with the bacteria. All right, any question about that? So the first two, generally more is better. The third, pH, generally is more acidic is better. And the fourth is generally better without the presence of organic matter. We can talk about the different types of disinfectants. And here I'm showing you all the different types of disinfectants. And there's a table talking about them. Let me go back and briefly talk about phenol and biphenolics. These are agents which are actually slightly toxic to humans, but they were the first disinfectants used in aseptic surgery. What's going on is both agents disrupt the plasma membrane and then start killing cells. Bioguanides also disrupt the plasma membrane, and these are fairly good antimicrobial agents, and we usually use bioguanides as skin disinfection, like when somebody's getting ready for surgical scrub. Halogens are things like iodine and bleach, which inhibit protein function and oxidize agents. And bleach is one of the best chemical agents for treating as an antimicrobial agent because bleach is readily available, it's cheap, and it's pretty ineffective. I'm meaning pretty effective. I didn't say that right. It is pretty effective at killing many microbes. It even kills some species of endospores, but it is not going to kill all endospores, meaning some endospore species will resist the killing by bleach. But vegetative pathogens are easily killed by bleach. Another problem, as I mentioned before, is that bleach is highly inactivated by other organic matter. Alcohol is actually a lousy disinfectant, and that's why we tend to use it more as a degerming agent, where it doesn't really start cleaning the skin when we're using alcohol. We call it a degerming agent, where it removes the microbes to safe levels, but there still are microbes after the alcohol application. Heavy metals, they're actually coming back in medicine because they can prevent microbial growth. And what we do is we put heavy metal on things like surgical wrap to help prevent the wound from getting infected. Soaps and detergents are used commonly in the home. And if you don't know, they're really lousy disinfectants. You only get about 10% of the cells killed with 1 unit of time. So you normally get 90% killed. So soaps and detergents are just not very strong agents. They do reduce the microbes, but they're not good at sterilizing. Any question about any of that? All right. Quaternary ammonium compounds are sometimes seen in the hospital. It's a much better surface active agent than the soaps, and we use them as an antiseptic for skin or instruments or utensils that other treatments wouldn't be too harsh on them, they would destroy them. So we can use quaternary ammonium compounds to help clean these items. And then there's chemical food preservatives. I don't think I'm going to be talking about these, but these are things we put in food to help preserve them. And that could be something like organic acids in cheeses, or nitrates or nitrites in meat products, such as ham, bacon, hot dogs, usually contain nitrates or nitrites. All right. Chemical sterilization cannot be done with most chemical agents, but there is one which does tend to kill endospores, and that is, actually there's two, sorry. There's ethylene oxide, which is a toxic gas, and you can put it on something and it will kill the bacteria on that exposed to the gas. It also kills the endospores, although you need a fairly long exposure to kill endospores. The other agent, which is good at killing endospores, besides ethylene oxide, a toxic gas, which isn't used very much because it's highly toxic to us. But if you remember, about in 2003 or something like that, somebody was sending anthrax spores through the mail. What they did was they took some of the material that had the anthrax spores and exposed them to ethylene oxide to kill off the endospores. Another thing they could have done, which they did not, was treat them to a peroxygen, such as hydrogen peroxide. These agents kill by oxidation, and they're very good at killing endospores. So hydrogen peroxide would be something that most of you have in your home, and they can kill vegetative bacteria as well as endospores. Any question about any of that? All right, comparing the effectiveness of various antiseptics, as I mentioned, soap and water is not very effective. After 2 minutes of time, you've only killed off about 90%, excuse me, that's the percentage of survival. You've only killed off about 10% of the bacteria. Most of us do not wash our hands for 2 minutes. All right. You can see Zephyrin has a little better killing than soap and water. Something that you usually find in your house is iodine or tincture of iodine. And as you can see, less than 2 minutes reduces the microbes down to only 10%. And tincture of iodine used to be very commonly used in the hospital. They would treat the patient's skin with tincture of iodine before surgery. The reason is you treat the skin with iodine, and that iodine sits there for a long period of time, killing for all that time. And then they will cut where the iodine is, and then the iodine also tended to stain the skin, so they knew where the iodine had been, where the bacteria numbers had been, well, almost not quite sterilized, but applying as an antiseptic. You can tell where it is. Any question about any of that? All right, let's talk about the last topic now, and that is different agents have different microbial characteristics, and we can rank them from the most resistant microbe to the least resistant. And this is a simplified hierarchy showing the relative resistance of the major microbial groups to common chemical biocides. The most resistant are prions. Prions are very difficult to kill. And we don't usually run into them, which is good because it'd be very difficult to remove prions. Typically, when they do surgery, they do not treat the surgical instruments to remove prions because you'd have to expose them to 1,000 degrees Fahrenheit to remove the prions. There is an experimental treatment where you could treat the surgical instruments to a proteinase, and that actually does remove the prions. And for when they think the surgical instruments have been exposed to prions, they will treat the surgical instruments with a proteinase to remove the prions. But usually all other antimicrobial treatments do not remove prions. The next most resistant would be the endospores. Most antimicrobial treatments do not kill endospores. If you recall, there were only 2 chemical treatments that did remove endospores, and that was peroxygen, such as hydrogen peroxide, and then ethylene oxide, toxic gases. All other treatments may not remove endospores. Any question about any of that? All right, mycobacteria is the 3rd most resistant. What happens with the mycobacteria? They have a cell wall that has lots of lipids in it, and that prevents most of the antimicrobial chemicals from getting in the cell. Curiously, well, let's talk about that in a little bit. The cystiprotozoa are highly resistant to antimicrobial treatment, but they're more susceptible than the mycobacteria or the endospores. The vegetative protozoa are the next most resistant. The gram-negative bacteria are the next most resistant. Fungi, including fungal spores, are the next most. Viruses without envelopes, the next most resistant. And then gram-positive bacteria are the next most. So gram-positive bacteria are actually sensitive to most antimicrobial agents. And why are they more sensitive than a gram-negative bacteria? It's because the gram-negative bacteria has an outer membrane on the cell wall, and that outer membrane can protect gram-negative bacteria to the antimicrobial treatment. That's something we mentioned before. And then the most sensitive would be viruses with lipid envelopes. You can denature the proteins, you can destroy the DNA, or you can destroy the lipid envelope, and then you make the virus non-infected. So the viruses with lipid envelopes are considered the least resistant of all of the microbes, the different microbes, to an antimicrobial agent. All right, I think I talked about that. I talked about mycobacteria, and then I mentioned that the endospores, very few things can kill them. Now, this general characteristics from most resistant to least resistant does not apply in all cases. Some of the characteristics of the bacterial groups, or of the microbe group, I guess I should say, can make them more resistant or least resistant to an agent. For example, I mentioned that alcohol is not a very good antimicrobial agent. And that's why we use it as a germicide instead of as an antimicrobial agent. Excuse me, we use it as a degerming agent, just to reduce the number of bacteria on our skin before we put the needle in, a degerming agent. But yet, alcohol is very effective at killing the mycobacteria. Why? The mycobacteria have a cell wall that has a lot of lipids. It's mycolic acid, if you remember that. And alcohol can go right through the mycolic acid of the cell wall. So the mycobacteria are very sensitive to treatment with alcohol. So alcohol, which is actually a very lousy antimicrobial agent, is fairly good at killing the mycobacteria. And the mycobacteria, for most other antimicrobial agents, they are resistant to most of those antimicrobial agents. So once again, you have to know that certain characteristics of the group, what those characteristics are, and whether it is sensitive to an antimicrobial agent. And that's the only one I'm going to mention. Any question about any of that? All right, I've already talked about prions. The only way really to completely kill them is to submit them to 1,000 degrees Fahrenheit. And that cannot be done on most things in the hospital, like surgical instruments, which can take a fair degree of temperature heating, but they can't take 1,000 degrees Fahrenheit, okay? And how do we kill them? Like I said, we haven't really come up with a good way. What they're doing is they're using proteinases whenever something is exposed to prions, and they cannot subject it to 1,000 degrees Fahrenheit. With the mad cow disease, with the cows in Britain, for example, they actually did incinerate them, expose them to 1,000 degrees Fahrenheit to get rid of them, to destroy the prions. All right, so while we have agents to control microbial growth, you should realize that the microbial methods are effective, but they are not uniformly effective against all microbes. For example, the endospores will be resistant to most microbial agents. The compounds discussed in this lecture are not generally useful for disease treatment in humans, with the exception of the antibiotics. Any question on any of that? All right, if there's no questions, that's it for this lecture. I'll say goodbye, and I will show up briefly at 6:30 to 6:45 to answer any questions. Goodbye.